Hello everyone, welcome back to my channel Capsa. In today's lecture, we are going to discuss a very important topic from microbiology and laboratory techniques, that is the autoclave. This is not just an exam oriented topic but something you will encounter repeatedly in laboratories, hospitals, and research environments. So, understanding how it works and why it works is extremely important. Let us begin with the basic question: what exactly is an autoclave? An autoclave is a sterilization device that uses moist heat in the form of steam under the pressure to kill the microorganisms, including resistant forms like spores. The name itself gives us a clue about its function. The word auto means self and clavis means a key or locking device. This tells us that an autoclave is designed as a self locking system capable of safely containing high pressure and high temperature during the sterilization process. Now, the next logical question is, why do we use moist heat instead of dry heat? The answer lies in the efficiency. Moist heat is much more effective and faster than dry heat. When steam comes in contact with microorganisms, it transfers heat efficiently and causes coagulation and denaturation of proteins inside the microbial cell. Once these proteins lose their structure, the cell cannot survive. This is the fundamental mechanism by which autoclaves achieve sterilization. Another important point here is that sterilization requires the presence of saturated steam. Simply heating without proper steam will not give the desired result. So, this brings us to the operating conditions of an autoclave. Under standard conditions, sterilization is carried out at 121 degrees Celsius under a pressure of 15 psi for about 15 to 20 minutes. In situation where higher temperatures are used, sterilization can be achieved at 134 degrees Celsius with 30 psi pressure in a much shorter time, usually 3 to 4 minutes. A very common misconception among the students is that pressure kills microorganisms. That is not true. Pressure only helps increase the boiling point of the water, allowing the steam temperature to rise above the 100 degrees Celsius. It is the temperature, not the pressure, that actually destroys the microbes. Now let us understand the autoclave physically by discussing its components and their functions. The pressure gauge allows us to monitor the internal pressure of the chamber so we know whether correct conditions are being maintained. The pressure regulating device ensures that pressure does not fluctuate beyond the required level. The safety valve plays a critical role by releasing excess pressure and preventing explosions, making the system safe to use. The steam release valve helps in controlled release of steam at the end of the cycle, while the vacuum release valve helps in removing trapped air, which is very important for proper steam penetration. The lead handle and the body handle allow safe handling of the autoclave, and the outer stand provides stability to the entire system. Once the autoclave is loaded, the sterilization process follows a specific cycle. First comes the preparation or loading stage, where materials are placed in such a way that steam can freely circulate. This is followed by heating or conditioning phase. In this phase, during which the steam enters the chamber and displaces the air, causing the temperature to rise gradually to the set value. After that, the sterilization or holding phase begins, where the load is maintained at the required temperature and pressure for a fixed period. Finally, during the exhaust and drying phase, steam is released, pressure returns to normal atmospheric levels, and the materials dry before removal. Now, where do we actually use autoclaves? So, autoclaves are extensively used to sterilize laboratory media such as agar and broth, laboratory glasswares like beakers, flasks, pipettes, and surgical instruments used in medical and dental procedures. They are also used to treat contaminated and biohazardous waste before the disposal, making it safe for the environment. So, to ensure that sterilization has truly occurred, quality control and monitoring are essential. Physical indicators like gauges and digital displays allow real time monitoring of the temperature and pressure. Chemical indicators such as heat sensitive tapes or stripes change in color when exposed to appropriate conditions. However, the most reliable method is biological monitoring using spore strips of Geobacillus stereothermophilus. If these highly resistant spores are destroyed, it confirms successful sterilization. That is why biological indicators are considered as the gold standard. Before ending, let us briefly talk about the safety precautions. Autoclaves should never be used to sterilize volatile or inflammable substances. Proper protective equipment such as heat resistant gloves and face shields should always be worn while opening the lid. One must always ensure that the pressure has returned to zero before opening the autoclave, and overloading should be avoided to allow proper steam circulation. So, students, this was a complete conceptual explanation of the autoclave, covering what it is, why we use it, how it works, and where it is applied. If this lecture helped you understand the topic better, please share it with your friends and subscribe to the CAPSA channel to support and grow this educational platform. If you want lectures of any topic, please write it in the comment section, and if you wish to contact me personally, you can do so through the email provided in the description. Thank you for watching and keep learning with CAPSA.